Hi, I'm a researcher at the NAM Research Group, and the purpose of this presentation today is to highlight our research interests and showcase the research capabilities of our lab. Our research is focused on the integration of quantum light sources into photonics-based processors with applications in quantum computing. To achieve this, we strain engineer two-dimensional materials. We have fabricated such samples which demonstrate this strain engineering. The two-dimensional materials that were used are hexagonal boron nitride and transition metal dihalogenides such as WSA2. The strain engineering of these devices affect their properties such as the wavelength of light emitted. Additionally, we have constructed waveguides as well that are capable of coupling the light emitted by these samples. We begin by observing these samples under the microscope, wherein we can see the features of the devices that we have constructed. Once we have observed the samples, we can characterize the light emitted by these samples in our characterization setup. In our setup, a laser beam is focused onto a sample in order to excite it. The photoluminescent emission from the sample is filtered from the reflected laser beam through the dichroic mirror and is then passed into the measurement module.
For our segment, we have three components. One will be the lasing component, the sample compartment, as well as the measurement compartment. Our lab consists of sensitive instruments such as the spectrometer, which enables us to measure the frequency spectrum of the sample emission. The frequency spectrum enables us to identify the wavelength of such sample photoluminescence emission, and also enables us to measure the variables that affects quantum efficiency, for example, intensity. Another instrument is the time-correlated single photon counting module, which actually connects to the single photon avalanche detectors over here. These two enables to measure the time correlation between the laser pulse emission as well as the sample emission. With this, we are able to measure the lifetime emissions of such samples, as well as we can further conduct experiments such as the Hanbury-Brown twist experiment or even the Hong-Ou-Mandel experiment. This allows us to measure correlation of two single photons or even the entanglement of single photons. From the lasing compartment, we have this pulse pump laser, which is fiber connected to the sample compartment. The pulse laser is first connected to this fiber collimator, where it passes through this series of mirrors onto a dichroic mirror, and it will reflect the 532 nanometers beam down this path onto this objective lens, converging the pulse laser onto a sample. The emission from the sample will pass back through this objective lens, through this mirror, onto a dichroic mirror, where it filters out the sample signal back onto this mirror and into this fiber collimator, where it will be passed on to the measurement compartment. In order to view the sample on the camera, we use a white light to shine onto the sample, which can be seen by a camera.
Our setup is highly modular. We direct a laser onto the sample to excite it and then observe its photoluminescent emission. The emission is collected into an optic fiber and is directed towards our measurement module consisting of a spectrometer and single photon detectors capable of measuring lifetime. The results can then be observed on the computer. Once we have measured emission, we can characterize it based on the spectrum on the spectrometer. Alternatively, we use the software PicoHub to measure the lifetime using the single photon detector. Lifetime is measured by first receiving an electrical signal from the laser module, informing the software when the laser pulse begins. After a pulse is sent, the single photon counter counts the number of photons received by it from the PL emission of the sample at different time intervals from the initial pulse. This data can then be used to obtain this plot consisting of an initial peak from the laser and then an exponentially decreasing emission photon count.